All right, so in this video, I'm going to walk you through the setup for the osmosis lab, where we're going to be finding out the tonicity of carrots, and you're going to be able to predict the calculations and relative concentrations of the solutions that we're working with. They're all going to be a mystery, and they're going to be color-coded from the start. Just a reminder that your Chapter 5 summary and Learn Smart, as well as your cell drawings from your textbook, are due today. And then on top of this, you have today's lab report. It's not a big report, it's just kind of interesting to go through. So you have your learning objective, which is still learners will be able to explain how cell size and shape affect the overall rate of nutrient intake and the rate of waste elimination, as well as learners will be able to make claims and predictions about natural phenomena based on scientific theories and models, right? We're doing a scientific theory and model today. So we're observing osmosis in living cells. So we'll be working with carrots because they are living organisms, or at least they once were before they were plucked from the ground, right? The interactions between selectively permeable membranes, water and solutes are important in cellular and organismal functions. For example, water and nutrients move from plant roots to the leaves and shoots because of differences in water potentials. Based upon what you know and what you have learned about osmosis, diffusion, and water potential in the course of your investigations, basically the last 2 diffusion labs that we did, think about these questions. What would happen if you applied salt water to the roots of a plant, and why? You want to think about what happens when water is going to move based on water potential, right? We know that pure water compared to salt water has a higher water potential, which means that water is going to move from where it's pure to where it's salty, right? But we know that plants are not pure water. So when you apply salt water to plants, different things will happen based on how salty you make that salt water. What are 2 different ways a plant could control turgor pressure, which is the pressure of when it's filled with water and the cell wall is not letting it burst, there's pressure, right? Is this a sufficient definition for turgor pressure? So plants also have this thing inside of their cells called a vacuole where they can store water, and they could purposefully fill the vacuole or push the water out of the vacuole to make sure that their cells are filled or not filled. Will water move into or out of a plant cell if the cell has a higher water potential than its surrounding environment? This is what you should know now after we did the water potential calculations. And you should know that when there's a higher water potential, that means that it's going to flow from high to low, so it's going to go wherever it is lower, right? So if there's a high water potential, but then there's another section that has a higher water potential, it's going to go from that higher to that lower, okay? Our solutions that we're going to be using are going to be 0 molar sucrose, which means that it's technically pure water. We're going to have a 0.2 molar sucrose. This is 0.4 molar sucrose, 0.6, 0.8, and 1 molar sucrose. So those are our solutions. They are color-coded, and you will not know which one is which or which one is which color for now, because you're going to predict those based on the results of this experiment. You should already know how to make the 1 molar sucrose, and making the 0.8, 0.6, 0.4, 0.2 is all similar because you're just multiplying the molar mass of sucrose by the molarity that you want. Originally, if it's 1, you would just add the entire molar mass in grams, but if you multiply it by 0.8, 0.6, 0.4, 0.2, you can create different molar masses that you're going to be adding to the same liter of water to create different concentrations. So in the procedure, technically I already prepped this for y'all because you can't do these, so this isn't a demo. I have 6 different cups labeled by their color. So I have a blue, a clear, yellow, purple, red, and a green cup, right? And they all have their different solutions, and all of the solutions are one of these 6 solutions. And I'm going to pat 6 carrots dry and mass them. And we want to record initial mass because we're going to be basically going over the percent change to decide which solution is which concentration. And then also we're going to be finding out what is the concentration of sucrose inside of carrots. Okay. After we record their initial masses, we're going to put each carrot in their cup, and then we're going to set them aside for 24 hours to wait for osmosis to occur. Once water has been moving for 24 hours, we will weigh them again to find the final masses, so then you can actually calculate percent change in mass. You're going to write a hypothesis about what you expect to see happen in a solution of high sucrose concentration. So when a solution is very, very sugary, right, 1 molar, for example, what will happen to the carrot? Will the water enter the carrot cells, or will the water leave the carrot cells? You're also going to write a second hypothesis about what you expect to see in a low concentration of sucrose. So we can talk about that 0.2 or that 0% sucrose, 0 molar sucrose, right? Will the water go into the carrot cells, or will the water leave the carrot cells? That's a hypothesis that you're going to write. After we collect the data, you will be graphing percent change in mass versus concentration of sucrose. I'll show you the graph setup on one of the following pages. And then you're going to be calculating, after you calculate percent changes, you'll be graphing it. And then we're going to be writing a conclusion. Your question for thought as you do this whole experiment is, how will you know which solution is isotonic to the carrot cells? Or basically, isotonic means the same concentration, right? We know that carrot cells have sugar in them. We just don't know exactly how much, so we'll find out during this lab. The hint is going to be in your graph once you graph. Okay, here's the formula for how to calculate percent change. I will put it on the pages following also, so if you need to copy it down, you can. So here is what your data collection is going to look like. You don't need to actually add the colors in. That's just personal preference to make it easier for me to differentiate them. But you're going to want a column of each of the cups, right? You're going to have a red cup, yellow cup, green cup, blue cup, purple cup, and a clear cup. And then you're going to want to leave a space where you can record the carrot's initial mass, which will be in grams. You want to leave a space for carrot's final mass in grams. And then your percent change in mass, which you're going to be calculating yourself using the percent change in mass formula. Okay. I will be recording the initial and final masses with you during this demo so that you can actually do the percent change calculations and the graphing and the conclusion on your own. So this is what you're going to end up with. So you're actually going to want to add this on the next page as well. So you're going to predict what the concentration of each color solution is based on the percent change in mass. So the setup is you're going to have the concentrations listed, right? I said before we have 0, 0.2, 0.4, 0.6, 0.8, and 1 molar of sucrose. And then you're going to be listing out your percent changes in mass. And then you're going to predict what color each one was. So basically you want to remember substances move from a high to a low concentration. And we're talking about water concentration right now. So as a hint, you will want to order your percent change in masses from highest to lowest to match them up from 0 to 1 molar sucrose. And then you want to have the attached color that was based on each carrot going with the color. So you can finally match up the color to the actual concentration of sucrose. The graph, as you can see over here, you're going to be creating your own graph title, and it's going to be y as a function of x as usual, right? And we know that earlier we said we're going to be graphing percent change in mass versus concentration of sucrose. So you are going to be telling me which one is the x and which one is the y in this graph. And the reason why the graph goes, the axis in the middle, is because some of these percent changes will be negative. And that is expected and it's supposed to happen. So if you don't have a negative number, that means you did something wrong. Okay. In your conclusion paragraph, you're going to be explaining, how do you know which color is which concentration? You're explaining the logic of how you found out the info in this table. You're going to explain for each individual color, right? You want to think about why the carrots gained or lost mass. You want to think about how much there was gained or lost compared to another one, right? That's just like when you're separating. If like 3 of them all gain mass, they're all going to be gaining different percents of mass though. What happens when the solution is hyper or hypotonic to the carrot cells? Right, remember hyper means that there's a lot of sugar and hypo means that there's less sugar compared to the water. When you graph your data, what does it mean where your data crosses the x-axis? Right, this is related to that question that I wanted you to think about during the entire lab, which is on this page in red. And then lastly, make a prediction what using a 2 molar sucrose solution would do to a carrot. So this is your conclusion paragraph that you're writing after you do your whole lab. And that's because you are doing this page, and then you're only doing the percent change in mass in this table because we're all collecting data together. So I'm going to actually do your initial carrot masses collection now, and then I'm going to post a separate video where we will do the carrot final mass because I have to give a 24-hour break between. So if we go over to our, see if the camera will show. Okay, so as you can see, we have the balance over here and we have our cup. We also have a selection of carrots that are around the same size, not exactly the same size, but it doesn't really matter because we will be calculating in percent change. So we're gonna mass each of them, and then I will be recording them with you to make sure that everybody has the correct data. So you wanna put the cup onto the mass balance first, and then you wanna zero it to make sure that you're not actually incorporating the mass of the cup with your measurement. So our first carrot we're gonna put in, and we're going to find the mass at 7.63 grams. So that means that on your data table, you want to write your initial carrot mass as 7.63 grams. And that one just so happens to be the one that we will plop right into the red cup. Okay, so that means that we are putting this one into the cup with the red sucrose solution. And I will add a little bit more to make sure that it is fully submerged afterwards. Okay. Our next one, we have to double check that we have it zeroed again. So after we add it, we want to click 0, and then we're going to add our next carrot in to see the mass of that one. So we have 6.63 grams, and that one we are just going to pop into the yellow solution. Right, so we're going to take this carrot, and we're going to pop this carrot into the yellow sucrose solution. Next carrot. .34 grams. And that one's going to go into our green sucrose solution. It's okay. Next carrot is going to be going into the cup for massing. .29 grams. And that one's going to go into our blue sucrose solution. Right, even though these solutions are like really heavily tinted, we're not really worrying about the color that the carrot is turning, unlike our last lab. We're just talking about the masses this time. Next carrot. .94. And that one is going into our purple solution. And then our last carrot is .09 grams. And we are going to pop that one into the clear solution. Okay. So now that we have our carrots inside of our solution, we're going to wait 24 hours. So you should have all your tables and graphs ready to go and ready and set up so that you can go ahead and take the rest of the data when we do our final mass video, and then our percent change in mass calculations. You're going to want to have your table for your pairing with your concentrations and your colors based on the percent changes in mass that you calculate, room for your graph, and then your conclusion paragraph that you will be turning in. So again, there will be one following video that has the results and the results only.